In this part of lecture number 10, I will cover passive and active transport. The cytoplasmic membrane in every healthy cell serves to separate the contents of the cell from the extracellular fluid. Without the membrane, there will be no cell, and therefore there will be no life. One of the main functions of the membrane is to permit certain substances to enter the cell and allow others to leave. Heavy traffic moves constantly in both directions through the cell membranes. Streaming in and out of the cell in endless procession go molecules of water, food, gases, waste, and many other substances. A number of processes allow for this mass movement of substances into and out of cell. These transport processes are classified as passive transport processes and active transport processes. The differences between the two categories is based on whether or not molecules of ATP is required to affect the movement of something through the cell membrane. Passive transport does not require energy of ATP molecules. It is a freebie for the cell. Cells do not have to pay with ATP molecules for substances to get in and out of the cell. Active transport requires cell to pay with ATP molecules for other type of substances to enter or leave the cell. How this works, I will explain later on in this lecture. First comes first. Let's talk about passive transport. Molecules have intrinsic kinetic energy, energy of movement. Therefore, they are in constant motion. The faster molecules move around you, the hotter it is. The slower the molecules move around you, the colder it is. Theoretically, there is such a condition as absolute zero. At this point, molecules do not move at all. It is denoted as 0 K Kelvin scale. But on the planet Earth, there is no such condition where matter does not move. Perhaps there could be such condition somewhere in space. Nothing can be colder than absolute zero on Kelvin scale or minus 459.67 Fahrenheit. So, as we established, temperature is determined by thermal motion of matter. Molecules have kinetic energy. All molecules are constantly moving inside us and everywhere around us. When there are lots of molecules in the area, it is known as high concentration of molecules. When there are only few, it is called a low concentration of molecules. Now, please pay close attention. First, remember one very simple rule, and later you will understand the whole concept. Here is the rule. Molecules tend to move from areas of high concentration to areas of low concentration. Did you see what happened? Look again. Molecules tend to move from areas of high concentration to areas of low concentration. Another way to say this, molecules move down a concentration gradient. This movement is a diffusion. Substances diffuse. Diffusion is a process resulting from random motion of molecules by which there is a net flow of matter from a region of high concentration to a region of low concentration. Or simply, diffusion is a movement of molecules from areas of high concentration to areas of low concentration. Diffusion occurs in gases, liquids, and solids. Diffusion is a good example of passive transport. By definition, diffusion simply is a process by which substances scatter themselves evenly throughout an available space. A good example of the diffusion of gases occurs when you open a bottle of perfume. There is a very high concentration of the perfume molecules in the bottle and a low concentration outside of the bottle. There is a high concentration of air molecules outside of the bottle and a low concentration inside the bottle. When the top of the bottle is removed, perfume molecules move out of the bottle and the air molecules move in the bottle. Each type of molecules move from area of high concentration to area of low concentration of its molecules. If you add crystals of potassium permanganate to a test tube with water, you can observe diffusion occurring in a liquid. It is easy to see the movement of substance in the water. The wispy tails of color are actually anions of potassium permanganate diffusing through the molecules of water. Of course, you cannot see anions themselves moving, only the color trails. Eventually, anions of potassium permanganate will be distributed evenly throughout the water. And if you added enough crystals and let it sit for over an hour, the whole body of the water is going to be colored intensively pink to purple solution. This is because anions move down its concentration gradient. In the lecture, I shared with you two of my water-soluble crayon paintings. I used diffusion technique, a spontaneous movement of color molecules from a high concentration to a low concentration. If you see closely, you will find some areas where you can see clearly how crayon molecules were moving from a high concentration areas to a low concentration areas. If you're not sure where to look, here is the most obvious areas. Some artists use acrylic powder to create these effects. Question to you. From your everyday observation, what environmental condition can affect the rates of diffusion? Think about it. Pause the video if you have to. You are correct if you say rates of diffusion depends on temperature and pressure. Think of cup of tea or coffee. You know that if you put sugar in hot liquid, sugar will dissolve faster than if you put sugar in iced tea or cold coffee. Why? Because in hot liquid, molecules move faster than in a cold liquid. Temperature is determined by how fast molecules move. If you would like to dissolve sugar faster in your drink, you would stir the sugar in a cup. Why? Because applying the pressure will make it dissolve even faster. So rate of diffusion depends on temperature and pressure. Now let me bring your attention to two definitions we are going to use in this lecture. Sometimes you will hear solvent and solute during this lecture. Solvent is a substance in which another substance is dissolved, forming a solution. And solute is a substance which is dissolved. In the picture, the solvent is water and solute is sugar. Let's get back to the cells. As you remember from the first part of the lecture, cell membranes are semi-permeable. A cell membrane allows some things to pass through while stopping others. For example, water molecules are small enough to pass through cell membranes, but large molecules such as sucrose cannot pass through cell membrane. Look at the diagram. What molecules will be able to go through the semi-permeable membrane shown on this slide? The water molecules. In what direction will be the molecules of water move? From solution A to solution B or from solution B to solution A? Pause if you have to think a bit. The answer is from solution B to solution A. This is because in comparison to solution A, solution B has more molecules of water. And as you remember, molecules move from area of high concentration to area of low concentration. Do you remember the definition of diffusion? Diffusion of water through semi-permeable membrane we call osmosis. So osmosis is a special type of diffusion. And since osmosis is diffusion, the water molecules are moving from an area of high concentration to the area of low concentration, and no additional energy is required for this process. Osmosis is a passive transport. So cell get water for free without paying with energy of ATP. Sometimes carrier molecules in a cell membrane can help in the process of diffusion. Carrier molecules can help move molecules across a cell membrane. This process is called facilitated diffusion. No extra energy is needed. Therefore, it is also passive transport. The environment that surrounds the cell determines the direction in which water molecules move. The amount of solute in the environment determines the tonicity of the cell. Tonicity is the ability of solution to affect the fluid volume and pressure in cell. When there are more water molecules outside a cell than inside a cell, water molecules move into the cell. A solution that contains more water molecules than located in the cell called hypotonic solution. We also say hypotonic solution, a solution that contains fewer solute concentration than the cytoplasm of the cell. You see, the less of solute in the solution, the more molecules of water in the solution, and vice versa. If a cell is placed in a hypertonic solution, a solution that has fewer water molecules outside the cells than inside the cells, water molecules move out of the cell. As the cell loses water molecules, it begins to shrivel up. In isotonic solution, the number of water molecules outside the cells is equal to the number of molecules inside the cells. When a cell is placed in isotonic solution, osmosis will not occur. Here I will repeat myself by putting already familiar to you diagram on the slide. As you already noticed, blue circle represent molecules of water and small red circles represent solute, any solute, could be ions of salt solution. On the middle of the diagram is the cell. So what kind of solution the cell submerge in? Correct answer is isotonic solution. The amount of water going inside the cell will be equal to the amount of water going outside of the cell. Therefore, the cell inside of this solution will be healthy and unaffected by the solution outside. But what is happening here? The cell is swelled up. Why? Because the cell is submerged in hypotonic solution. There are more molecules of water on the outside of the cell than inside of the cell. Because inside the cell, there are more solutes. Solutes replace the molecules of water. So water molecules from the solution outside are rushing into the cell, making it swell. This can lead to the rupture of the animal cell and cell death. What is happening on the last diagram? The cell is shriveled. Why? Because cell has less solutes on the inside, meaning more water molecules inside and less water molecules on the outside. So water molecules are rushing from the inside the cell to the outside. That is why when you are getting ready for dinner, you do not want to put dressing on the salad before all guests arrive. Otherwise, your salad will get soggy. What has more molecules of water? A glass of seawater or a glass of fresh water? Answer is a glass of fresh water. There is because the ions and minerals of the seawater displace the molecules of water. In comparison of seawater with fresh water, which is a hypotonic solution and which is a hypertonic solution. In this example, the seawater is a hypertonic solution and the fresh water is hypotonic solution. This is because seawater has more solute and therefore less solvent, water molecule, compared to the fresh water. If you compare two glasses that are both filled with seawater, those solutions will be isotonic to each other, since they will have the same amount of both solvent and solute. Students in general biology classes often have lab on osmosis. They make dialysis bags that are made of semi-permeable membrane. Large molecules such as sucrose cannot go through the membrane, but water molecules can easily travel in and out of the bag. On the diagram, you see such bags. Before submerging them into the different solutions and beakers, students first weight them and record the data in the data table. Then at the same time, they place the bags into the beakers with different solutions. And then 10 minutes after, they take them out all at the same time and weight them again. Let's now think. What will happen to the weight of the bags after 10 minutes exposure to our different solutions? Look at the bag number 1. Will the weight of the bag increase, decrease, or stay the same? To answer this question, you need to determine, is the solution in the beaker hypertonic, hypotonic, or isotonic? You are correct if you say it is isotonic. This means that the amount of solvent and solute in the bag and in the beaker is the same. Therefore, the weight of the bag will stay the same if there was no hole made by an accident. There will be no net movement of water towards the solution or the cell. The cell and the environment around it are in equilibrium, and the weight should remain unchanged. Now, how about the bag in the beaker number 2? What will happen to the weight of that bag? Of course, it will stay the same because the solution in the beaker is also isotonic. Same amount of sugar and water in the bag and in the beaker. How about the bag number 3? Where is more water, in the bag or in the beaker? You're correct if you say in the beaker. There are less water in the bag because there is 20% of sucrose, table sugar solution in there. So, will water rush outside the bag or inside the bag? Of course, it will go inside the bag. Remember, molecules move from area of higher concentration to the area of low concentration. So the weight of the bag will increase. How about bag number 4? Yes, the weight of the bag will increase because there are much more molecules of water in the beaker and much less molecules of water inside the bag. So, now what is going to happen to the last bag in the beaker with 60% of sucrose? You are correct. The weight will decrease. The water will rush out of the bag. If this was an animal cell, the cell will shrivel. I hope you understand how osmosis may affect tonicity within the cell. If not, you will have to go to the beginning of the lecture and watch it over again. Sometimes one has to use the vocabulary before grasping the concept. Both diffusion and osmosis are types of passive transport. The cells do not have to spend ATP molecules to get things in and out of the cell. Such substances as water, oxygen, carbon dioxide, lipophilic compounds go easily through the cell membrane. They diffuse through the cell membrane. However, cell membrane restricts diffusion of hydrophilic compounds, large molecules of sugar, amino acids, and highly charged particles. The passage of such molecules and particles relies on the specific transport protein embedded in the cell membrane. And for such transport to happen, cell has to pay with ATP molecules. This is what we call active transport. Active transport is a movement that requires energy. Sometimes molecules must move against the direction of diffusion in order for the cell to reach homeostasis. This also could be done by active transport. Cell has to spend ATP molecules for this to happen. The sodium-potassium pump is used by nerve cells to maintain the proper levels of sodium and potassium ions in the cell. The pump transports sodium and potassium ions across the cell membrane in such a way that homeostasis is not reached. This allows electrical charges to be created across the cell membranes that enables the impulse to move along the nerve cell. Cells are also able to bring in and excrete molecules that are too large to pass through the opening of the cell membrane. Endocytosis is the process that allows a cell to bring in materials from its surroundings. There are two types of endocytosis. Phagocytosis, cellular eating, allows cell to engulf large particles. Pinocytosis, cellular drinking, allows a cell to take in liquid molecules. Pinocytosis is accomplished when the cell membrane falls inward, pulling the dissolved molecules in with it, as forms a depression in the membrane. Later, the opening to the depression is closed, trapping the liquid. The bottom of the depression breaks off to form vacuole in the cell. Exocytosis is opposite of endocytosis. Exocytosis allows materials to be discharged and secreted from a cell. Exo means out. For example, proteins produced in the ribosomes of the cell are transported out of the cell by exocytosis. In this second part of the lecture number 2, we covered different types of passive transport, such as diffusion, osmosis, facilitated diffusion. Also, we briefly looked at the examples of active transport. In our third part of the lecture, I will introduce you to different types of cell organelles and their functions.